In this video we will discuss about CAR-macrophages sources. So far in our previous videos we have discussed about CAR T cells and CAR T cell sources, and also we have discussed about CAR-NKT cells and CAR-NKT cell sources. And here we are discussing about the CAR-macrophages as another alternative to fight with the tumors. So before going into the different sources of macrophages, let us understand the two main categories of the macrophages. This M1 like macrophage and M2 like macrophage. So the macrophages that acquires the M1 like phenotype have the pro-inflammatory, and they can kill the tumors through phagocytosis. And these are some of the receptors that are found on the M1 like pro-inflammatory macrophages, whereas M2 like anti-inflammatory macrophages has a completely reverse characteristics. That is, it helps in the tumor growth, immune evasions. So our objective is to get the M1 like macrophages, because these are the ones that can help in the killing the tumors. So what are the different sources of these macrophages that are currently available? One of the sources is through this peripheral blood, from which we can use the cytokines, this GM-CSF, to differentiate it to the M1 like macrophages. The other source is about this induced pluripotent stem cells. From there it can be differentiated using the multiple steps to this M1 like macrophages. And there's one cell line also available, that is THP1 cell lines, that can also be used to differentiate to make this M1 type macrophages. The one thing that we need to be careful while using these cell lines is that the cells needs to be irradiated. Now the M1 like macrophages needs to be irradiated before we can use it for the treatment, to avoid any possibilities of tumorigenesis because of the contamination. And so once these CAR-like macrophages are formed, they can kill the tumors through phagocytosis. Whenever they come in contact with the tumors, it will release the phagocytosis will be triggered. And at the same time it will also release the cytokines, pro-inflammatory cytokines such as interleukin beta, interleukin 6, TNF, and that will not only helps in killing the tumors, but it will also helps in the activations of other immune cells such as T cells, NK cells, dendritic cells. And these M1 type macrophages could be very useful in the tumor microenvironment, as they have the more they have the more capacities to enter inside the tumors or infiltrate the tumors. And we can see that it can act even on this M2 type macrophages, so that the tumor growth and tumor invasions could be suppressed. And at the same time we can modify the CAR macrophages in a way that it will help in the trafficking, and we have understood the modifications of the receptors that helps in the trafficking in our previous videos. That is, the trafficking are usually helped by these homing receptors. Homing receptors helps in the trafficking of the immune cells at the site of tumors, or at the site where it is most needed. So this way it will also improve the anti-tumor efficacies of the CAR macrophages. And finally the paper suggests that these three approaches could be used in combinations so that they will have a synergetic effect. These three combinations means the CAR type macrophages, or the CAR macrophages, CAR T cells, CAR-NKT cells, and these three can be combined together to have a synergistic anti-tumor effect or response against the tumor. So that will greatly improve the CAR-T diagnosis efficiencies against the solid tumors. So that's all for this video. I hope you understood the different sources of the CAR-macrophages and different types of the macrophage. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.